All right, so we get to talk about chemical synapses. And so we've got four outcomes for today for this little mini lecture. So let's just jump right in. So a synapse, this is the synapse right here, this space in between a neuron and another cell. It can be another neuron, it can be a muscle cell, it can be an endocrine gland, like there's lots of things. Okay, but that's a synapse, is this space. Okay, so we have what's called a presynaptic neuron, and then we've got the postsynaptic neuron. The easiest way to tell the difference between which one is which is that the presynaptic neuron, when you see it, it's going to be the one that has the vesicles. The postsynaptic neuron is going to have the receptors. Okay, now the presynaptic neuron could also be, if there's another neuron up here, so that's my neuron, okay, this would also, up at this point, we would consider this the presynaptic neuron. So it's not either or, it just depends on what you're referring to. So we look at two neurons at a time. One's going to be pre, one's going to be post. Okay, so a chemical synapse has several things. It has voltage-gated channels. And okay, those are here. And oftentimes, not always, because it's not shown on this picture, but you'll just see them as like little parallel lines like that. Okay, you've got the synaptic vesicles, okay, which are, I don't know where my little marker went, so we'll find it. There we go. So synaptic vesicles, like it says, it's pointing to these circular containers that have the neurotransmitter. We've got a reuptake molecule that's not shown, but that's something that'll take this neurotransmitter and bring it back into the cell. We've got a receptor, which will be on a ligand-gated channel, okay? So right here. And then we've got an enzyme, again, not shown, but it's going to break down the neurotransmitter, and that's going to be important. So when a neuron wants to talk to another neuron or even another cell, how does this work? Well, we're going to break it down into pretty simple. Okay, so we have an action potential, and this action potential comes all the way, from wherever it starts, and it comes down. It always goes down towards the axon. Okay, action potentials only travel one way. That way, a presynaptic neuron is always talking to the postsynaptic neuron. Okay, when that happens, it causes a depolarization that opens up voltage-gated calcium channels. This causes calcium influx. So why is calcium going in? Well, the reason is due to flow-down gradients. We've got lots of calcium out here, so we're going to write it big. And we've only got a little bit of calcium inside, so a little bit. And so due to flow-down gradients, it goes in. Well, why do we have calcium go in? Well, it's essentially the signal or the green light for exocytosis to occur. And so calcium will flow into the cell and cause exocytosis. So exocytosis, there's two parts. One, the vesicles will move. Okay, and so we see that with step 5. And then we have the actual release of the neurotransmitter. Okay, and so pokes the hole or whatever, and the neurotransmitter is released, and it diffuses. Okay, because of flow-down gradients. There's lots of neurotransmitter inside the vesicle, not so much in the synapse. And ultimately, that neurotransmitter could bind to a receptor. Now, it's not necessarily going to. Okay, it's not like there's a magnet that pulls in, or a tractor beam, or whatever you want to say, that pulls the neurotransmitter from the vesicle or from the presynaptic neuron to the receptor. That neurotransmitter can go anywhere. It can go any direction. And if it happens to run into a receptor, that's great. Okay, and we refer to this as a chemical synapse because one cell is communicating with another via a chemical. Okay, there are electrical synapses that we will not talk about in this class, but that's also very important. It's essentially how your heart is able to contract. All those muscle cells fire at the same time because of what's called an electrical synapse. Now, once this happens, we don't want neurotransmitter just floating around forever. We need to remove it. So, but why? And the reason is, well, this neurotransmitter is basically the stimulus for something to happen. So let's just say that you're doing a bicep curl and you've got neurotransmitter that's released, and it tells your muscle to contract, so you have bicep, so elbow flexion due to bicep contraction. Well, if you don't get rid of that neurotransmitter, you're going to have a contracted bicep for a long time. And so we need that to go away so that the muscle can relax, and then you can contract when you want to do another bicep curl. Now, there are 4 ways that this can happen. Okay, so if you look at this picture, hopefully you can identify all 4 of them. So the first, and here they are. So I'm just going to go through them from left to right. So one, the neurotransmitter can be taken up by another cell. Don't worry that it says glial cell here. Just think another cell. Two, remember that reuptake molecule? It can just diffuse or be taken back into, diffuse is the wrong word, but it can be taken back into the presynaptic neuron through a process called endocytosis. It's essentially the opposite of exocytosis. It can just diffuse away into the bloodstream, or there can be an enzyme such as something called acetylcholinesterase, which breaks down a neurotransmitter called acetylcholine. And notice that we go from neurotransmitter here to broken neurotransmitter there. Okay, and so we need this to happen so we don't have constant stimulus, and it allows us to have another action potential or communication or whatever you want to call it when we're at a time when we're ready for it. So what do you need to know? Be able to identify the pre- and postsynaptic neurons in a diagram, describe how one neuron communicates with another cell, and explain why we need to remove neurotransmitter from the synapse.